Here we go, my friends. The parasympathetic nervous system, the answer to the complexity of the sympathetic nervous system. That was a tough one. That was like that workout where you never wanted to work out at any point in time. You dreaded started. You did it because you knew you had to. You hurt from the moment you started. It was just agony, but hopefully you felt better afterwards. That's what the sympathetic video was. This one's going to be a breeze. Parasympathetic for two reasons. One, at each effector, they're mostly the same effectors as a sympathetic. If you know what the effect is at the sympathetic, by the sympathetic branch, go to the same effector and just swap it. If it increased secretion via the sympathetic arm, it's going to decrease secretion via the parasympathetic, or vice versa. So that's easy. And for two, the outflow is much, much simpler. There's not those 12 ways, right? Sympathetic nervous system is like asking somebody for directions and them giving you 12 ways to get there. It's like your brain fell off, right? Much simpler here. There's only 5 roots, and they're really pretty simple, and they're not too complicated, and we'll work through that easily. And then second, test yourself. You'd come here and you just knock these out, and you would avoid the ones in red because there is no parasympathetic innervation to the ones in red. There's sympathetic innervation to those, but you can just skip them for parasympathetic because there's no specific innervation from the parasympathetic arm of the nervous system. All right, here we go. Let's just get this out of the way. Again, we're monkeying around in the autonomic nervous system. We're just about to finish it. Next video, we'll move over to the enteric, but now we're just knocking out the parasympathetic arm, or the parasympathetic division of the central nervous system. And it is rest and digest, or rest and recover. Just calm down. You escape from the bear. Life goes on. Now figure out all you're trying to do when you were running away from that bear. Let's all agree you got out of whack. You got every system in your body pretty much far away from equilibrium or homeostasis. Let's go back and engage all our homeostatic mechanisms and get back to homeostasis and get metabolizing again. That's the big deal with the parasympathetic, because you're not metabolizing anything while you're running away from a bear. It's got its own feet. So now we'll start with the sensory, right? Sensory, integrate, motor, always the same that we take. What is the sensory? Well, it's got its own feed of information. And again, it's going to come in on 3, 7, 9, and 10. Cranial nerves 3, 7, 9, and 10. And it's going to feed in, and it is slow sensory information. There are times in your life where you have a quarter of a second to a half of a second to live or die. You're on the freeway, you lose concentration for a split second, you see brake lights in front of you, and you're going 80 miles an hour. You have a quarter of a second to decide if you're going to live or die, right? This is not that. That would be sympathetic. This is parasympathetic. Your stomach, you ate too much, you're getting a little stretch. Your stomach's going to send some, hey guys, tone down on the food a little bit here. And it's a slow pathway. Your blood pressure's creeping up a little bit. Some slow sensory information via those 4 cranial nerves, kind of telling you what's going on with 2 types of interoceptors, right? Internal receptors, or visceroceptors, visceral receptors. 2 things. One, stretch. How much is are the walls of the blood vessels stretching? How much is the stomach stretching? How much of the intestine stretching? And then chemoreceptors. What is the partial pressure of oxygen in our blood? What is the partial CO2 right now? What's the blood pressure? It's just lots of things. What's the chemical composition, the osmolarity of our blood? Things like that aren't life or death immediate situations, and they need to be sensed, but they follow what are called slow sensory pathways via those 4 cranial nerves into the central nervous system. That's their path into the central nervous system. And then what is the integration? Well, you've got your Pane, again, going back to the pneumotaxic and the apneustic respiratory centers, right? You've got all those are all known. And you've got your vomiting and your hiccuping, and it sounds weird to say, but your sneezing and your coughing centers. Those are all autonomic centers, so you have integration there. Sacral level, you're going to get back to micturition. You're going to get back to wanting to urinate and defecate under parasympathetic innervation once the fear of being eaten by a bear is over. Your limbic system, which is that sort of fear and rage and aggression and all that's calmed down. That's not impacting your parasympathetic. Leave that for the sympathetic, right? It's not impacting your parasympathetic that heavily. And of course, in the hypothalamic, we're now moving away from the anterior. Now we're moving to the posterior nucleus. The posterior hypothalamic nucleus is a huge one. It is involved in autonomic, and it is driving the parasympathetic. It's sending upper motor neurons down the line, and it's going to ping on the cranial and sacral cell bodies, because we have cranial sacral motor output, and that's what we have right here. Okay. The sympathetic was fight or flight or fright. I don't even know why I have that on here. Give me just one moment, if you would, please. I have no idea why that's on here. I forgot to change something in a slide. We are not interested in sympathetic. This is a video on parasympathetic. No fight, no flight, no fright. This is happy times, okay? Rest and digest and recover, right? Sorry about that, typo, okay? And on the motor side, you are sending, just to refresh here, you're sending cholinergic, myelinated, preganglionic neurons, and those are all in solid red here. Preganglionic, they're all cholinergic. They're releasing acetylcholine at the ganglia, and they're all myelinated, and they're all just cruising along. And just to sort of reiterate that, here's the picture. So these are the ones that I just showed you coming out of the lateral gray horn on the anterior root. Thousands of times we said it, but it's worth saying it. Myelinated, preganglionic, cholinergic neuron, because it is releasing acetylcholine. Now on the postganglionic, two things of interest, right? Still cholinergic, but now it's unmyelinated, and it's going to the effectors, and it's going to cause the opposite effect that the sympathetic nervous system had on those same effectors. And the outflow is, of course, coming out of the cranial, these four cranial nerves, 3, 7, 9, and 10. So the hypothalamus, the posterior nucleus of the hypothalamus is pinging these four right here. I'll just show it kind of pinging these four, but I'll show the top two, but it's running all the way down, and it's pinging these three down here, the sacral S2 to S4, right? And that's how you're going to accomplish parasympathetic motor action at the effectors. But I want you to take a look and just be a little perceptive of the lines and just follow the lines. Here's a preganglionic motor neuron. It runs to the ganglion. These are called terminal ganglia, right? Terminal ganglia. But they're also called intramural ganglia too. And watch these. Take a look at oh nope, not that one. Take a look at the vagus nerve. Look at the length of the preganglionic neuron. It is running all the way to the actual organ. There's no synapsing here. The preganglionic neuron is not synapsing at a sympathetic trunk ganglion. It's not synapsing at a prevertebral ganglion. It is running all the way to something called an intramural. What does intramural mean? It's not just sports in college. Inside the wall. Inside mural is wall. Inside the wall. And so the preganglionic neuron, I'm following this one right here, is running all the way in. Actually, let's follow this one. Follow this one here. It's running all the way in. I think this gives you a good view. Yeah. To the stomach, and it is synapsing. The ganglion is in the wall of the stomach. And then look at the size of the post. I'll circle the post-ganglionic neuron. Look at the size of that post-ganglionic neuron. It is tiny because it's already in the wall of the organ. So what can we say in general about the pre-ganglionic neurons and the parasympathetic? They're going along. Remember we said they were short in the sympathetic? They just went mostly right, usually in the idealized model. They went right from the spinal cord to the sympathetic trunk ganglia. They were right next to it, literally just lateral to it. Short pre-ganglionic neuron, and then maybe a longer one to the effector. Here, you want to look at the other way. Super long preganglionic all the way to the effectors, to the intramural ganglion, and then a super short postganglionic neuron. Just something to think about. Okay, couple paths we just have to iron out. No big deal. Let's just crush these. Path number A, you're coming out of cranial nerve number 3. So your posterior hypothalamic nucleus is pinging right here and synapsing on the nucleus of oculomotor nerve 3, cranial nerve 3. It's running out to preganglionic neuron. How many times have we seen this? Running to the ciliary ganglion, synapsing there. Running along a post-ganglionic neuron to the eyes, and it is getting to the ciliaris and the intrinsic muscles of the eyes. Not the extrinsic, not the skeletal, the lateral rectus and the medial rectus, and the superior rectus and the inferior rectus, not those, but to the smooth muscles of the eye, like the ciliaris. And what is it doing? Again, I'm just going to verbalize these. It is constricting the pupil, and it's closing the pupil so that you don't have your eyes wide open like dinner plates. You're no longer running away from a bear. You're chilling out, and you're just going to constrict the pupil. and your accommodation reflex will be towards near vision. You'll accommodate towards near vision. Maybe once you got run away from the bear and you're safe, you want to open up a can of corn and enjoy some delicious corn. You might need to read something on that. Or you want to make a recipe and you might need to read something. Whatever. Okay? A calm response. I have no idea where that came from, but you can follow along with what I just said, presumably. Okay. Let's put in cranial nerve 7. We knew this was a big one. So now you're pinging the nucleus of cranial nerve 7. You're sending motor outflow out, and you're going to head to a ganglion called the pterygopalatine ganglion. Where do you think that one is? Right? Remember the pterygoid processes of the sphenoid bone? Right near the palatine bone, right in that little interesting area. Come down through the foraminal valley, and you kind of pop up right there. Great place for a ganglion. And you run there and you synapse, and then you send out your postganglionic nerve. And now you're feeding a whole bunch of glands in the face. Not the parotid, save that for just a second, see how that's coming off a separate innervation. But all these glands in the face you're feeding. Okay? Lacrimal glands, nasal glands, palatine glands, and a couple of salivary glands, but the sublingual and the submandibular. And what are you doing? All of these, you're now going to increase secretion, right? You're going to start cranking up secretion. Time to create some tears. Maybe you got a ton of dust in your eyes while you're running through the forest for your life, and you want to wash that out. Nasal gland, get nasal secretion going, get mucus secreting again. Palatine gland, salivary, get salivating sublingually and submandibular. Get ready to eat, get ready to digest, get ready to get metabolism going again now that you're sure your life is saved and you did not get eaten by that bear that we've now talked about probably 50 times right now. Okay, and the last one on this, so cranial nerve number 9, and this is sort of the last one that's following, going to the head of the face, right? So glossopharyngeal cranial nerve number 9, coming out, again, pinging the nucleus, running out on the preganglionic neuron. Now you're going to the otic ganglion, okay? The otic ganglion. And then from there, you're running on your postganglionic unmyelinated neuron, to the parotid gland, and that's the big daddy. That's the parotid gland. When you get that salivation and you feel that tingle when you just smell delicious food, that's your parotid back there, and it's firing up, and that's just a jet of saliva into your oral cavity, right? You're just cranking that through the roof, and you're going to start salivating to facilitate chewing up the food, digestion, the food in the mouth, and increase all those glands too that you're going to secrete, all the fluids you're going to secrete, and you're just getting ready to digest and metabolize. That's all. But that's only what I just talked about. A through D were really only about 10% of the output. 90% of the motor output is, of course, coming through the granddaddy of them all, the wanderer, the vagus. Okay, and that is feeding off of cranial nerve 10. Here's your very long preganglionic neurons, all the way to the intramural ganglia, the ganglia in the walls of the organs, and then these really short postganglionic. I hope the picture is showing you what I'm trying to say. Really short postganglionic neurons inside the walls of the organs. Okay? And what are you serving? You're serving the viscera, right? Just read it, read it, read it, read it. Esophagus, heart, bronchioles, liver, gallbladder, bile duct, stomach, small intestine, pancreas, colon, kidney, and your. That's all you have for viscera. That's the who's who of your viscera right there, okay? And it's feeding all of those, and it's telling them all to calm down. So what's the effect at all of these? At the esophagus, you're going to increase, so I say calm down, but you want to increase the digestive side. So you want to increase mobility at the esophagus. Start swallowing and moving food down into the stomach. You want to constrict the bronchioles. No more need to suck in all the air in the world and just suck in air like you're heaving, okay? Constrict the bronchioles, start breathing normally. Heart, the nodal or the pacemaker cells, down with the rate. Drop the heart rate back to normal heart rate. Stomach, small intestine, colon, increase mobility, start moving food through. Increase secretion, get your amylase, get your bile duct going. Next one, bottom one, get your gallbladder and your bile duct, get your fat digestive enzymes in there. Get your proteinases in there. Get all your enzymes in there. Start breaking apart that food and start increasing absorption at the small intestine. Right? Start pulling in all those good nutrients and rebuild your metabolic state. At the colon, get mobile and start moving the fecal matter through. At the pancreas, okay? Zippo on the glucagon. Kill the glucagon. We no longer need glucagon. We do not need blood sugar high. In fact, we want to go the opposite way. So now at the pancreas, you are going to what? secrete insulin, the one we know about. And insulin is going to go into your blood. It's going to pull the sugar out of your blood and get it in your cells, right? There you can metabolize it. You can get back to a good metabolic state by undergoing cellular respiration and just kind of getting back homeostatically to a better metabolic state, assuming that your run from the bear took you out of a good metabolic state and kind of stressed you out, right? Liver, you're going to start glycogenesis. You're going to take all the glucose that's individual monomers, like units and like cars on a train, and you're going to couple them together into a huge train called glycogen. And you're just going to store it for the next time a bear runs after you, okay? Gallbladder, bile duct, increase secretion of bile duct, increase the action of the biliary tree, get more bile into there to emulsify fats and help to digest yummy fats and give you that great metabolic energy from fat. Renal parenchyma, start producing urine again. Start filtering through the glomeruli, start producing urine. It's time to urinate again. It's time to defecate again. And the ureters start increasing mobility so that you can increase your urination and defecation and get back to homeostasis, void the bladder, void the colon, right? And just get back, void the rectum, get back to homeostasis, okay? Good metabolic health is what we're talking about here. And finally, if you go all the way down, you want to come all the way down the line to the sacral part of the cranial sacral outflow. Everything I've talked about so far has been cranial. This has come out of cranial nerves 3, 7, 9, or 10. Now you come down to the sacral outflow, and S2 to S4. Follow path E. And where are you headed with that? Okay, long preganglionic neurons, same concept. And now you're hitting these really short intramural postganglionic neurons. Okay, where are you headed? You're headed to the rectum. You're headed to the colon. You're headed to the urinary bladder, to the kidney, and to the male and female external genitalia. And what are you doing when you get there? At the descending and the sigmoid colon, sorry, missing an O, my bad there. Descending sigmoid and descending in the sigmoid colon and the rectum, increasing mobility. Pushing the fecal matter through to defecate. Get that going, okay? Urinary bladder, start contracting the detrusor muscle. Start emptying the urinary bladder, right? Release the constriction on the urinary sphincter so that you can urinate. And at the male and female external genitalia, you are looking at erection. So erection of the penis and erection of the clitoris too are part of the parasympathetic. A little bit odd, a little bit off topic maybe, but they are part of the parasympathetic, and that's the innervation and that's the effect at the male and the female external genitalia via parasympathetic innervation. And here's the table. Read it and celebrate. Don't read it and weep. Read it and celebrate, because this has everything. Great study guide right here. Sympathetic on the left, the thoracolumbar. Parasympathetic on the right, the craniosacral. This is everything that I've just said for the last 50 minutes, 55 minutes on this subject. It's all right here. Use this as a review. Every single thing you could possibly want. It even rolls on to the next page. So specifically at some simple, I don't have them all listed here, but just some random effector organs. I don't have them all right here, but some random effector. And uterus, we've talked about a couple of these, but hair follicles. So this is kind of important because I want you to show, I want you to see something like this. So hair follicles, erector pili, and the muscle, and also all the other parts of the integument. Here's your effect sympathetically, but then there's no innervation. And that's why you're going to see those things. You're going to come up red on the test yourself. Okay, so let's just knock this out and be done. We're looking directly at the oculomotor coming from CN3, cranial nerve number 3. And that's going to be sensory output to the smooth muscles of the eyes. Okay, preganglionic neuron running to the ciliary ganglion, and then from the ciliary ganglion synapsing there and running a postsynaptic neuron to the intrinsic or the smooth muscles of the eye. All right, facial nerve is going to be preganglionic neurons running motor up to the pterygopalatine and the submandibular ganglia. And then synapsing there with postganglionic neurons that are then serving the lacrimal, nasal, and the palatine, and the salivary glands that are not the parotid, so the sublingual and the submandibular. Glossopharyngeal, on the other hand, will be serving the parotid, and that's its key role, is to run the preganglionic neuron to the otic ganglion, and then run the postganglionic neuron from the otic ganglion to the parotid gland and start squeezing the parotid gland, the big one, and just saliva jets streaming into your mouth. Right? Cranial nerve 10 from the vagus, you are going to get A. sensory input from the viscera first, but then your preganglionic motor output is going to the intramural ganglia, so that long preganglionic neuron going to the intramural ganglia of the esophagus, the bronchioles, the heart, the stomach, the small intestine, the colon, the pancreas, liver, gallbladder, bile duct, kidney, and ureter. Any questions there? Pretty straightforward, right? And then S2 through S4 are going to serve, they're going to become the pelvic splanchnic nerves, and they're going to send motor output to the intramural ganglia in the colon, the descending in the sigmoid colon, and the rectum, urinary bladder, and then the male and female external genitalia. That is that. And then we come here. Do not answer the red because there is no parasympathetic innervation. Cross those all out. But simply go to where you looked at what the sympathetic innervation was and just flip it. Okay? What was, I'll just do the first one. I'll do 1, and I'll do 8, and I'll do 15. I just literally picked the 3 most random numbers I could think of, except I started with 1. At the eyes, what is this sympathetic effect? You dilate like wild the ciliaris, and you accommodate for far vision, and you try to bring in as much light and far vision as you possibly can as you're trucking through the forest full speed being chased by a bear. What is the parasympathetic? The complete opposite. Constrict the pupil, drop down the light, accommodate towards near vision, things right in front of you, and just take it easy and relax and oppose the effects of the sympathetic. 8, at the liver, we wanted to crank up sympathetic. We wanted to crank up glycogenolysis. Glycogen, the compound, the macromolecule, lysis meaning cutting. So we wanted to crank up glycogenolysis. We wanted to crank up on the sympathetic arm. We wanted to start chopping glycogen into glucose so we could feed it into the bloodstream. Glycogen is a great storage molecule, but it's the glucose monomers, the individual units that we want. And so we just start chopping, chopping, chopping. What is the opposite parasympathetic effect? Now we start glycogenesis. We completely reverse the process. We start pulling glucose into the liver, and we start polymerizing it. We start making long chains of glycogen and storing them away for a better day. All right? For a rainy day, so to speak. 15, the rectum. Under sympathetic innervation, you just calm the rectum down and you stop mobility. Just shut down mobility. Peristalsis. Shut it down. What is the parasympathetic effect? Crank it back up again. Start creating peristaltic action in the rectum. Start moving fecal matter through so that you may defecate again. Right? Get back to that sort of homeostatic balance. I'll let you fill in the rest. If you have any questions about it at any time, just ask me and I can go through these all with you if you'd like. But I'll leave that to you. And thank you so much for sticking with these couple beastly videos. This one I told you was easier, 12 minutes shorter. The enteric is going to be a breeze because it's just the gut brain. We'll be through with it and done. We'll be on to neurotransmitters. We'll actually be done with chapter 15 in 2 more modules. Chapter 16 will be a breeze on the sensory and the motor systems. And we'll cruise right up to the special senses in chapter 17 and finish off this whole nervous system binge that we've been on to start off this course. Thanks, and I really hope that helped a lot.